Today we're going to talk about carnosine. What is carnosine? Carnosine is a dipeptide. A dipeptide is a molecule that is made up of two amino acids. And you can see the peptide bond, which makes up that amide functional group that connects the two amino acids. On the right, we have the amino acid histidine. On the left, the remaining portion comes from beta-alanine. So those are the two amino acids that make up the dipeptide molecule carnosine. It's composed of histidine and beta-alanine. Now perhaps you heard of the amino acid alanine. What's the difference between alanine and beta-alanine? Alanine is a 3-carbon amino acid. Like most amino acids, it has a carboxylic acid functional group and it has an amino group. Now the key here is to notice the location of the NH2 amino group, and that is it's on the alpha carbon. The alpha carbon is 1 carbon away from the carboxylic acid carbon. Well, beta-alanine, like alanine, has 3 carbons. It has a carboxylic acid functional group, but the difference is it's not a typical alpha amino acid. The NH2 group is on the beta carbon. So that's why it's called beta-alanine. So it's a beta amino acid. There's also GABA, gamma aminobutyric acid, where the amino group is on the gamma carbon. Now, out of the 2 amino acids that make up carnosine, beta-alanine is the rate limited factor. We'll call it the RLF. So what this means is that the rate at which carnosine is produced in the body depends on how much beta-alanine is available. Now let's talk about some of the functions of carnosine. Number 1, it acts as an antioxidant. What does that mean? It means it reduces the overall oxidative stress in the body. It works as a free radical scavenger. It can basically neutralize free radicals and other reactive oxygen species that could be produced through cellular respiration. Now, carnosine is also an antiglycating agent. What does that mean? So this means that it inhibits glycation. Glycation is the process where sugar can bind to protein, and in such a way it can alter the shape and function of that protein. And this occurs through something known as the Maillard reaction. Now this has to be a reducing sugar. So the carbonyl group of a reducing sugar reacts with the amino group found in the amino acids that make up protein. Now, lysine is especially susceptible to this reaction because in its R side chain it has a primary amino group which is available to react more than the other amino groups in amino acids. Most amino acids, the NH2 group is found on a secondary carbon, whereas in lysine, the NH2 group is on a primary carbon in its R side chain, and that makes lysine especially susceptible to this reaction. So perhaps if you remember from organic chemistry, whenever you have a carbonyl and a primary amine, it's going to form a Schiff base, which is basically an imine derivative. So this is how reducing sugars can react with, this is how they can bind with protein, is by reacting with the amino groups found in those protein molecules. Now, this is just the first step of a series of reactions that occur after this, including rearrangement, glycoxidation. You also have polymerization in some cases, and even dehydration reactions that follow this, creating a series of other compounds known as AGE, advanced glycation end products. And this creates all sorts of other problems in the body. So this is one of the dangers of having excess sugar in a diet. This increases the risk of glycation, where those reducing sugars can interact and bind with those proteins, altering their shape and function. And not only that though, but those bad proteins now can cross-link with other proteins that are functioning properly. So now you have this cascading effect that can occur when there's excess reducing sugars in a diet. Glucose, fructose, those are reducing sugars. Sucrose is not a reducing sugar. However, when it breaks down into glucose and fructose, it breaks down into reducing sugars. So here's a study that talks about some of the benefits of carnosine. When you think of the word carnosine, the word carn means flesh, in that it's prevalent in meat like chicken, beef, fish, and things like that. But notice its role as an antiglycating agent. It says carnosine is useful in helping prevent damage caused by too much sugar in the body. Sugars, such as glucose or fructose, can bind with proteins. This binding not only damages the protein, but the glycosylated protein can stimulate secondary inflammatory processes. Glycosylation of protein is one of the four major destructive pathways stimulated by oxidative stress in the body. The binding of sugars to protein can also lead to proteins sticking to each other, a process known as cross-linking. And you could read the rest of this article as well. Now, there's another one that talks about glycation and AGE, or advanced glycation end products. And you could see the carbonyl group of glucose reacting with an amino group, which is found in all proteins. And we can read this here. It says AGE were initially identified in the cooking process as a result of a non-enzymatic reaction between sugars and proteins within foods. This reaction is called the Maillard reaction. The glycation process is initiated by a chemical reaction between the reactive carbonyl group of a sugar or an aldehyde with a nucleophilic free amino group of a protein, leading to the rapid formation of an unstable shift base. And we can see that here, where the aldehyde of glucose or even the ketone group of fructose, it can react with the amino group of an amino acid in proteins, creating a shift base or an imine derivative, and that leads to a series of other reactions. And so you get AGE, or advanced glycation end products, and it just creates a whole sort of other products that can have different effects in the body. But notice lysine is especially involved in these reactions. You could see lysine binding with these sugars and creating these different molecules. So lysine and arginine, because of the NH2 group that they have in their side chains, they're susceptible to the Maillard reaction. Now, the mechanism by which carnosine acts as an anti-glycating agent, scientists have yet to figure out. But some researchers believe that the primary amino group found in the beta-alanine portion of carnosine interacts with the carbonyl group of reducing sugars to form shift bases. However, because of the histidine part that's attached to carnosine, particularly the imidazole ring, it is believed that these shift bases are stabilized, thus reducing their capacity to further degrade into these advanced glycation end products. So here is another article that talks about this. It says, although the mechanism by which carnosine inhibits the formation of AGE remains to be elucidated, it is likely that the free amino group derived from beta-alanine competes with the amino groups of proteins in a reaction with reactive 1,2-dicarbonyl compounds. However, beta-alanine itself has a much smaller age inhibitory effect than carnosine, and the neighboring imidazolium moiety of carnosine shows synergistic activity in the stabilization of Schiff bases derived from carnosine and 1,2-dicarbonyl compounds. So even though the amino group of carnosine can react with the carbonyl group of reducing sugars, it is believed that the imidazole ring from the histidine part of carnosine could stabilize the Schiff bases and kind of prevent them from degrading further. And going to this part, says, thus carnosine competes effectively with the amino groups of lysine residues to form Schiff bases, acting as an effective age inhibitor. Further, in the presence of carnosine, the ability of methyl-glyoxyl treated ovalbumin to form cross-links to a second protein, alpha-crystalline, is dramatically reduced, showing the carbonyl sequestering ability of carnosine. So just to keep it simple, carnosine works as an antiglycating agent, which is very useful. And oxidation also accelerates the formation of these age products. And what's interesting here is noted that vitamin C, E, and A are all effective radical scavengers, and they attenuate glycoxidation, thereby leading to reduced formation of age. So what does this all mean? Even though excess sugars in the body can accelerate the formation of these age products, oxidative stress also plays a role. Vitamins A, C, and A are antioxidants. They can reduce oxidative stress. Thus, they play a role in reducing the formation of these advanced glycation end products, as well as carnosine. So the combination of carnosine and these vitamins can greatly reduce the formation of advanced glycation end products. Now, carnosine also has other functions. It can also chelate heavy metal ions, and it also works as a buffer, stabilizing the pH. It has a pKa of 6.8. I won't go too much into that, but hopefully this gives you a good overview of how carnosine works in the body and its benefits